Everyone, it's Mr. Sinti, and today I'd like to talk to you about reading the genetic code, or if you will, cracking the genetic code. And it is kind of like a secret or a secret message, and the message, as you may know, is in the sequence of nitrogen bases in DNA and both in messenger RNA. But deciphering the code wasn't altogether that easy because one of the problems that scientists had in the early 1960s was how can you possibly read such a code? How do we know what A stands for, or U stands for, or C stands for, or, in other words, if you had, for example, let me pull this up here. Say you had a piece of messenger RNA like this. Go messenger RNA. And let's put out the nitrogen bases like this. So say you were a biologist in the 1960s and you had to try to decipher the code. Let me just put something out there like that. I'll go C, G, A, U, G, G, C, C. Okay, how do you decipher the code? See, it's a little bit difficult, and you're like, well, I'm not even sure what the question's asking. Like, when you say decipher the code, in other words, do you recall like the structure of an amino acid? Here's a typical amino acid. Here's the amino group. Here's the alpha or central carbon. This is 20 different things coming off, and here's the carboxylic acid. So there's 20 of these amino acids in the cytoplasm or in the cell, and what we're trying to do is, let me erase it like this, and what we're trying to do is try to figure out the sequence of amino acids. And so do you see that R group that I left remaining there? There's one of 20 different kinds. And so if I were to draw an amino acid, make it a little bit more simple and say, let's go here, let's say that an amino acid, I'm just going to, instead of drawing the R group, I'm just going to call them number 1, number 2, number 3, number 4, all the way up to 20, right? So these go all the way up to 20. 20 different amino acids floating around. So how does the ribosome know what the correct order is? Because do you recall that the sequence of amino acids, it's critical, it's sort of like spelling a word. In other words, which one is it? Does number 17 go first? Does number 20 go first? It's a bit of a dilemma. You're like, well, I don't know. You said earlier that it's the nitrogen base sequence that codes for the amino acids. That's how the genetic code is read. I did say that. It's certainly not phosphate, sugar, phosphate, sugar, phosphate, sugar. It has to be the sequence of nitrogen bases. And so how does it work? Well, at first, scientists thought, well, perhaps the nitrogen base A stood for one of the amino acids. Like, in other words, maybe it's number 1. I'll put it right there. And maybe uracil stands for number 2. See how we're making a little protein? Guanine stands for the amino acid number 3. And see, this is how we roll. And then cytosine stands for number 4, and uh-oh. See how we're kind of in a stuck right here? In other words, if there's only 4 nitrogen bases in the genetic code, and this goes all the way to 20, and if there's 20 amino acids, how in the world can we code? In other words, there's 20 students in the class, we only have 4 names. It's not going to work. So clearly, the genetic code isn't read one for one, and you're like, well, maybe it's read in, let's see, let me erase this. Maybe the genetic code is read in groups of 2. What I mean by that is, say it's AU break, and that stands for amino acid number 1, and GC break, that's the second amino acid. It's this way as the third one. You're like, well, this may make more sense. But if you know your mathematics, you'll know that if there's 4 nitrogen bases and you read them one at a time, like we did first, there'd be 4 possibilities. Obviously, that doesn't work. What we're doing here is reading them in doublets. So that's 4 nitrogen bases read in groups of 2. So what are all the possible combinations if you read this? 16. That doesn't look like it's going to work either. And so this is a bit of a problem that biologists faced. And so let me draw your attention to this right here. This is a little animation showing us the same kind of problem that we that we have. So when you open up a bag of protein, it's assembled into all 20 different amino acids, and so there they are. These are the three-letter acronyms for the 20 different amino acids. But see, here's the problem. In DNA, there's only, same thing with messenger RNA, there's only 4 different nitrogen bases, but you get 20 amino acids. And so that's a problem. And so at first, maybe it's like read 1 for 1. You're like, that's not going to add up to 20. That's probably not the way it's going to work. You're like, oh, let's do 2. So as I was showing you before, even if you read it in groups of 2, it still only codes for 16, and you have 4 remaining. And so yes, you guessed it, the genetic code of all of life on the planet Earth is read as a triplet. 3 bases at a time code for 1 amino acid. 3 bases at a time codes for 1 amino acid. So this is the secret code. It's called a codon. Every third 1, 2, 3 amino acid, 1, 2, 3 amino acid. Now, if you're following the mathematics on this, you know that we kind of overshot it a little bit. We went a little bit overboard. We're reading the 4 nitrogen bases in groups of 3, which totals 64. And that's going to altogether be not too bad, because when we have a lesson on mutations, we'll see how this can be an advantage. And so that is how the genetic code is read. It's read in groups of 3. And so, of course, there's more groups of 3 than the 20 amino acids, but you get the point. And so I think that's kind of a cool thing. So as we come back here, let's talk about, in our class notes, let's talk about how the genetic code is read. And so first thing I want to say about the genetic code is that if it consisted of a single nucleotide, there wouldn't be enough. There'd only be 4. And if you read it in a group of 2, it wouldn't be enough. It'd be 16. So the genetic code is read in groups of 3. And so here's the DNA. And so what you can see here is each group of 3 is called a codon. So it's 1, 2, 3, break. 1, 2, 3, break. 1, 2, 3, break. So here's the sequence along the gene, right here. Here's the gene of DNA. And obviously it goes out further. And that corresponds to the amino acids below in the polypeptide. So this is how the genetic code is read. And so it's in triplets of nucleotides. That's the smallest unit of the code that will code for all 20 amino acids. So it's a triplet code. So there's 4 raised to the 3, which is 64, which means we overdid it. But basically, the DNA codes for, in the sequence of nitrogen bases, the amino acid sequence in the polypeptide in 3-letter words. So I really like this picture here. Here's DNA. In dark blue, there's 1 gene, and let's say that that's an intron right there. And here's another gene right here in yellow, gene 2. If we break that down, now remember, let me sort of draw a little bit here. Remember, DNA isn't really directly responsible for making a protein. It's sitting in here in the nucleus. I can sort of enclose this as well. But what happens is messenger RNA is produced or transcribed, and so this is important. So as we said that the 3 bases is the code, what we're really operating with is messenger RNA. So the genetic code to make a protein is really carried by messenger RNA into the cytoplasm at the ribosome. That's where the genetic code, in other words, the sequence of nitrogen bases, is translated into a polypeptide sequence. So translation is reading the genetic code. Let's check it out. Let's go here, a little bit more narrow. So do you see how this is uracil, guanine, guanine? So it's UGG, and then we'll break it up like that, and then U, break it up. So these are the triplets. These are the codons. And so this corresponds to one of the amino acids, the other one, the other one, the other one. So here's the big question. You're like, okay, I can see that a codon translates to an amino acid, but here's the big question. I'm not going to answer it right away, but how did they decipher this? I mean, it's one thing to understand how the code is read, but how do you know, just by looking at UGG, that it's tryptophan? or UUU is phenylalanine. How do you know that? It's a good question. It was a Nobel Prize winning experiment that came up with that. So during transcription, in other words, the making of RNA, messenger RNA is transcribed from DNA, and so that's what contains the genetic code. The complementary nitrogen base sequence of messenger RNA is the genetic code for making a protein. And so, same sort of picture. Here's the gene in DNA, and then here's the gene in messenger RNA, and then each three are a codon, which make the protein. And so during translation, in blocks of 3, the codons are sequenced into a polypeptide sequence. So during translation, the codons are read, and each codon codes for one of the 20 amino acids that will be put into the polypeptide. And because of the fact that there's 3 nucleotides for every 1 amino acid, that means that it's going to take, for example, 300 nucleotides to make 100 amino acid long protein, because it's 3 nitrogen bases for every 1 amino acid. So let me just sort of ask you a question about that. Like, say for example, we had a protein that was 200 amino acids in length. So here's a protein, and it's like 200. I'm not going to draw out all 200, but there's 200 amino acids in length. How big is the nucleotide in messenger RNA? In other words, how many nitrogen base sequence would code for that? And the answer to that would be 600, because it's for every 3 corresponds to 1. Okay, and so in the early 1960s, there was a great geneticist by the name of there's a great scientist by the name of Marshall Nuremberg, and Marshall Nuremberg and colleagues deciphered the genetic code. Very clever, and basically what they determined was this is the special decoder and how to decipher it. And so they worked this out. For example, like if you had a piece of, let me draw this, of messenger RNA, and this is most important. You got to remember that it's messenger RNA that we're deciphering. And so if you had something like AUG was the very first codon. Let's go something like CCC. Let's make it GUU. I'll just make 3 amino acids. And so as it turns out, every protein they learned began with the codon AUG. So there it is, AUG, break. And so the amino acid met. So I can put this up here. Go a different color. So blue for the amino acid met. So there's the amino acid met. And then the next one coming in would be CCC. You're like, well, how does this work? First base would be cytosine. The second base is also cytosine right there. And the third one is also C. So it's CCC, which means it's the amino acid proline. So that's the next one, proline. So are you following this? Let's do one last one. GUU. GUU would be the amino acid valine. And so that one would go there. And you're like, well, this will continue on forever on the ribosome. No, not necessarily, because as it turns out, there's 3 codons which are called stop codons, and that means the protein's finished when you reach this special sequence of 3. And so this is how the genetic code is deciphered. And so basically the sequence of nitrogen bases in DNA and in messenger RNA is unique. So each gene is the recipe for each different protein. And so that's pretty cool. There are thousands long nucleotides because the proteins are really long. And basically when you have these 4 bases, you can put them in any possible combination, so they can make up any possible protein combination as well. It's almost limitless. And so the linear order of bases in the gene corresponds to the linear order of amino acids in the protein, and that's the primary structure. In other words, what amino acids, what order of the amino acids and which ones are present. And then ultimately that'll determine how the protein folds, or its 3-dimensional folding or conformation, which then ultimately determines its function, like sort of like different shapes of a key. And so in our textbook, we use this kind of a decoder. So let's try this. Let's play a little bit of this and come up with our own little sequence here. And if you can do this, you're good to go in reading the genetic code. So let's start off with AUG, and then let's make up something. Let's go GGA, or CCU, and then let's go, how about CUA? And then let's go UAA. So can you decipher this? In other words, can you translate the nucleic acid sequence of messenger RNA? I know it's RNA because you can see it's uracil here. So what you would do to solve this is, can you do this? Break it up in thirds, like this, or codons. So there's 1, 2, 3, 4. This one's going to be a stop codon, I think. So check this out. So you look at your decoder and you're like, what is the amino acid sequence? We'll make it over here together. So it's A, which is the first letter right there, and then U. First, second, so AUG is methionine. Yes, we knew that. So let's go black for the amino acid. So it's MET. That's the first amino acid. You're like, what's the second amino acid going to be? GGA. Let's go G. GA is glycine. Oh boy, glycine. Right there. And the second one, third one, CCU. CCU. Oh, it's proline. Notice how CC anything over here is proline. Because remember the genetic code was 64, so we overshot the 20 amino acids. But I'll come back to that point. So this is proline. So CCU is proline. How about CUA? CU, and then A is leucine. Leu. And then finally UAA. So it's UAA. Hey, a stop codon. So that means we're finished. Stop. And so we can go like this. It's a stop codon. So that means there's no more amino acids and the protein's finished. Now obviously a protein's bigger than 4 amino acids, but I don't think you would like us to go any further. And on this note, stop codon, we're also done talking about the genetic code. So we'll stop there. I hope you enjoyed watching the video. Thanks.